In this lecture, we will discuss the connective tissue, blood. Part 1, we will look at the composition of blood and red blood cells. Blood is a type of connective tissue. It's a complex mixture of cells, chemicals, and fluids. Here, the definition of connective tissue comes because blood, even though it's fluid, it connects different parts of the body. Therefore, it is considered a connective tissue. Blood transports substances throughout the body, and it helps maintain a stable environment within the body. It also helps in heat distribution. Blood transports nutrients from the gut to various parts of the body, and it transports oxygen from the lungs to different cells, and then it removes metabolic waste, including carbon dioxide, and bring them either to the lungs or else to the kidneys to be filtered out. Blood, heart, and blood vessels altogether make up the cardiovascular system. Blood includes red blood cells, white blood cells, platelets, and plasma. Of these four components, three components are solid structures, whereas plasma is the liquid matrix. So red blood cells, white blood cells, and platelets are called formed elements because they are cells, solid structures. When we look at blood cell types, there are many different types and we'll look at them individually. And when it comes to characteristics, students are expected to know the characteristics and how to identify, differentiate, and functions of each types of these blood cells. So we have an abundance of red blood cells. We have then these ones that are with an asterisk. They are all white blood cells. Neutrophils, lymphocytes, monocytes, basophils, and then we have platelets, which are these parts of cells, small cell components. When we take a blood smear, which is you take a blood sample and thin it out, spread it out on a particular slide, and on that you will be able to see numerous red blood cells. And as you can see, there is this characteristic light region in the middle and darker region on the outside. Then you have these larger cells. These are the white blood cells, also called, or the most common component are neutrophils. And then you have these parts, fragments of cells, and those are going to be the platelets. We talked about blood is composed of four different components. Three are elemental forms. One is the liquid form. To be able to separate the cells from the liquid component, we will be able to obtain blood and do something called centrifugation. Here what you do is you put the blood in this particular tube and you spin it down at a fast speed. When you spin it down, the cells separate from the liquid. The cells all precipitate down, and we will see mostly red blood cells. You'll see a little bit of white blood cells on top, and then the liquid form. So this is how you are able to separate the plasma from the red blood cells, or from the blood cells. An adult blood volume is about 5 liters. So as you can see, it's not a lot of blood, but that is the blood that circulates throughout our body and helping maintain and a stable inner environment. Again, blood composition, we talked about how we will have the plasma and the blood cells. Plasma is a mixture of water, amino acids, proteins, carbohydrates, lipids, vitamins, hormones, electrolytes, and cellular waste. And 55% of total blood is about plasma, and about 45% is erythrocytes, red blood cells. You have less than 1%, which will include white blood cells and platelets. So erythrocytes, when we looked at the blood smear, I showed how the erythrocytes had a a more lighter middle component compared to the outer side. That's because erythrocytes have this donut shape like inner indentation. If you look at a cross section, you'll see almost like this dumbbell shape. The reason why it is like this is these red blood cells have to go through small capillary tubes, and the cells are much larger. So this indentation allows the red blood cells to fold and even pass through capillary vessels and small vessels. So when you see this type of shape, know that these are the red blood cells. And it's called biconcave disc. So it's concaved on both sides, having the biconcave or the donut shape. So now we'll talk a little bit about the production and control of red blood cells. Production and control of red blood cells. So red blood cells are produced in the bone marrow of these long bones. We talked about how they have cavitaceous structures in the long bone. The red bone marrow will be there, and that will produce red blood cells. So this is controlled by this negative feedback mechanism, where when the body realizes, oh, we don't have enough oxygen, or we need to be able to carry more oxygen, the kidney will get the signal and it will produce a compound known as erythropoietin. Erythropoietin goes through the blood. When it reaches the bone marrow, erythropoietin helps helps drive the production of red blood cells. So when there is more red blood cells in the body and it's able to carry more oxygen, now that inhibits the production of erythrocytes. So that's the negative feedback mechanism. Shown here the same thing in a different angle, where the body will realize, oh, we don't have enough oxygen, and then the kidney and sometimes even the liver will produce erythropoietin, and erythropoietin will go through the blood, reach the red bone marrow, produce more red blood cells that will help carry more oxygen, and that more oxygen will now stop the production of erythropoietin. What happens if there is more erythropoietin, more red blood cells that are produced, more than what we need? So an excessive increase in red blood cells is called polycythemia. Polycythemia causes our blood to be very viscous and start moving slowly. And that, since the blood is now moving slowly, even though there is more red blood cells, that can cause oxygen deficiencies. So polycythemia, we will get this reddened skin because there is more red blood cells, and it will also start having this itchiness on it. And the treatment for this type of disease is going to be regular blood draws. Like for blood draws for donation, we can draw the excess blood and then help the blood to flow more smoothly. So hematopoietic stem cells, these are the original cells where all the blood cells differentiate or are made from. So when we see some of these cells that don't have a proper shape, where the nucleus is almost round and almost the whole shape of the cell, these are hematopoietic stem cells. These can differentiate into forming the different types of cells. Now, we will not see these cells floating in the bloodstream. These are present only in the bone marrow, because bone marrow is the place where maturation or differentiation of blood cells happen. So the hematopoietic stem cells, the original cells, can differentiate into several different cells, forming monocytes, T lymphocytes, erythrocytes, a cell known as megakaryocytes, and neutrophils. So of these examples given here, erythrocytes are the red blood cells, megakaryocytes are the cells that produce platelets. So we talked about how platelets were fragments of cells. So these large cells give out fragments that become platelets. Then the other three cells that we have, monocytes, lymphocytes, and neutrophils, are part of the white blood cells. So all of them originate from the hematopoietic stem cells. Life cycle of a red blood cell. So we talked about how red blood cells are produced in the bone marrow of long bones, and they circulate in the blood. And after a certain time, they tend to break down, they tend to not have integral function. Liver, as blood passes through the liver, is able to detect this, and the liver will catch cells, liver and even the spleen will catch cells that are not doing well, and they will recycle these cells. They will break these cells down, and components of the cell will then be used again to form new cells. And of this, there is a component known as bilirubin, where the hemoglobin groups are broken down. So this bilirubin and then will be put into um, the gut, to the intestines, where that can either be recycled or eliminated. The average lifespan of red blood cells is 120 days, so about 4 months in circulation. We will next look at white blood cells and platelets in the next lecture.